Okay, but anyway, folks, at any rate, in all seriousness, what activates this pathway here? Okay, well, most of these things are activated by these molecules called growth factors. And what are growth factors? Growth factors are molecules that stimulate things like cell division. That's why we call them growth factors. Now, a little story behind this, a little historical introduction to growth factors. A long time ago, people were trying to culture human cells especially, and animal cells and stuff like that. At first you think, okay, get a petri dish, fill it up with all kinds of nutrients and goodies, just like you culture bacteria, right? And then throw these cells in. So they did that. What happens? The cells cringed up into little spheres, they rounded up, they sank to the bottom of the dish, they didn't divide, their metabolism was extremely slow, and eventually they died. They certainly did not behave like normal cells. And people kept on trying to do these kinds of things, growing vertebrate cells and human cells in culture, and they just didn't have any luck. They tried all kinds of nutrient solutions and what have you. And then there was this woman who had, I think it was an ovarian or cervical, I think it was a cervical cancer in Baltimore back in the early 1950s, a woman by the name of Henrietta Lacks. Her doctor was also an active researcher. He decided to try to collect some of these cells. She had an extremely aggressive cancer. In fact, it killed her several months later. And he figured, well, these cells are so fast growing, maybe I can somehow coax these guys to grow in culture. So he took some samples from biopsies from her and tried to grow these things in culture. Well, at first it didn't work, but then he came up with the idea, okay, well, these cells in the body are being exposed to body fluids, blood, stuff like that. Maybe there is something in, like blood serum or whatever, that is necessary for these cells to behave normally. So he decided, let's get some blood serum. So he went out and got some blood serum from horse or something like that, and threw some of that in the culture dish, and lo and behold, those cells started behaving normally. They started attaching to surfaces, things like that. They started proliferating, so he's able to grow these cells in culture. In fact, nowadays they're called HeLa cells. They're epithelial cell line, and they've been used up, down, left, right, and center labs. I used to work with them myself a long time ago. Okay, now, and ironically, if you took all the mass of all the HeLa cells in the world currently out there in various labs, it would probably outweigh her hundreds of times to one. So her legacy is still left on, even though she died decades ago. Her cells are still there and stuff. And they do grow very aggressively in culture. Okay, well, figured, okay, there's these things in serum, and nowadays when you grow cells in culture, you have to add blood serum in various amounts from animals. It turns out if you add them from fetal animals, like fetal calves and stuff like that, that's more effective than adding from adult organisms. So what were these mysterious things that were in the serum that were necessary for cells to proliferate and behave properly? Well, people start calling them growth factors. And once again, in biochemistry, the term factor refers to something that does something and they have no idea what the heck it is yet. Okay, so we started calling these growth factors. Of course, many years of research started identifying what these things were. And what these growth factors are they're small secreted proteins. Various cells, most cells produce and secrete numerous different growth factors. I'll just call them GFs for the time being. Different cells produce different growth factors. Many cells produce many different kinds of growth factors. Others produce a more limited thing. What they produce and secrete can vary depending on how far they develop. So a cell at one point in differentiation may release certain growth factors and then stop doing that and release something else later on. Okay, now the other thing is each cell responds to specific growth factors. Most cells respond to several different kinds. Some respond very strongly to certain kinds and not so strongly to others. But each cell responds to various kinds of growth factors. And what they respond to can also change as the cell develops and differentiates. So what they produce and what they respond to can vary with time. Okay, now, most of these things are produced by cells. Growth factors in general tend to have a fairly local effect. Now, they do get in the bloodstream and stuff, but their concentrations are often fairly low. But their strongest effect is fairly close to the source tissue. In other words, a place that's actually secreting these growth factors, they'll have by far the strongest effect in cells that are close by. And different concentrations of these growth factors can have different effects. One of the big problems with stem cell biology is even if you know what growth factors you need to use to stimulate differentiation, you have to use the right ones at the right concentrations. Too little, and cell may go another way and differentiate another way. Too much, same thing, you may get a different differentiation pathway. The right amounts, the right concentrations, the right time, and the right duration of exposure to these. So when you try to turn a stem cell into a fully differentiated cell, and we've only been able to do that step by step with a limited number of cell types, you have to hit them with exactly the right growth factors, right concentrations, right time windows, and in the right order to get the cell type you want. If you mess that up, you'll get some different cell type, or maybe some cell simply won't survive. So, have all these different kinds of growth factors. Now, over the years, people have identified numerous growth factors. Many of them use the RAS-MAPK pathway, but many of them use other pathways instead. So not all growth factors use the same pathway. As a matter of fact, there's one pathway we'll mention that a specific large family of growth factors use. There are several others that we're not going to mention, but many of them use this RAS-MAPK pathway. So let's take a look at examples. Many of these are families of proteins. One of the widest used ones are a family of growth factors called the EGF, or Epidermal Growth Factor family. It was first called that because it helps stimulate the production, the differentiation of the dermal cells into, in other words, lower layers of skin, into epidermis. Now there are also a couple other growth factors, especially ones that generate the very most outer layers of the skin. But the EGF family does that, but it's involved in stimulating differentiation of many mesodermal type cells. So we're not just talking about making skin, we're talking about a whole bunch of other different types of cells and tissues that use members of the EGF family. Many different members of this family, widely used in all kinds of developmental events, tissue repair and maintenance and stuff like that. Okay, another big family, the fibroblast growth factor family. Let's make sure we can spell it right. Now, fibroblasts are cells that produce things like cartilage and stuff like that. So they're very active secreting cells. If you take a look at a tendon or a ligament or something like that, you'll find living fibroblasts inside them producing that material. And it was first discovered because some members of the family stimulate fibroblasts to produce and secrete the extracellular matrix that forms the connective tissue, and it stimulates their proliferation as well. But FGF is used in a wide range of different kinds of developmental events, especially things involving what we call mesodermal type tissue. Now think of this. FGFs are involved, for instance, in different types, in certain aspects of limb formation. Limb formation depends on several different growth factors being secreted from certain centers and stimulating growth. You look at a limb, okay, you have to have an axis from the base to the tip, you have a top and a bottom, or dorsal and ventral, and you got a left and a right surface. All three of those axes are controlled by secretion of certain members of various growth factor families that cause cells to differentiate in one way here and in a different way there where the concentration is lower or higher. So we use these guys quite a bit. Other examples. Hepatocyte growth factor. Hepatocytes are liver cells. And livers have an amazing ability to regenerate. They can take a real hammering and still come back and regenerate themselves. Think of this. If you were to donate liver tissue to somebody for a transplant, they'll generally take about half of your liver out. And that might save somebody's life. Within 6 months, your liver is completely grown back again. Pretty impressive. Yes?
Is there a limit as to how many times, like, if you take out half and it regrows half?
Yeah, there's a reason for it, that cells have tend to have a limit in how many times they can divide before they peter out. That's what we call telomeres, and telomerase can restore those. Yeah, so you couldn't do that indefinitely. But when you think about, okay, good case in point. Let's suppose you have somebody who is a big-time heavy drinker, right? You know, the kind of people like you see at the frat houses that down a 12-pack a day, and let's say they continue doing that. Okay. You know, let's face it, alcohol, especially in large quantities, is fairly toxic to the liver. Okay, so it's going to start killing off liver cells. They will generally regenerate, but if you keep on doing it over and over and over again, and you drink a 12-pack every day for 20 or 30 or 40 years, eventually some of those liver cells won't properly be able to regenerate. The liver tissue gets replaced with fatty tissue and connective tissue and stuff like that, and then your liver function starts to decline. That's what we call cirrhosis of the liver, and that's a major common thing for serious long-term alcoholics. A lot of them basically trash their livers. Don't believe me, go out to Russia. I don't think anybody in Russia has a liver left anymore. They love vodka. It's like mother's milk to them. Okay, but at any rate, yeah, actually it's a serious problem. Now, aside from the effects of alcoholism being way up on the premature death thing in Russia, alcohol poisoning, meaning literally drinking yourself to death in one city, like occasionally happens at frat parties, is in the top 10 causes of death. A friend of mine brought back, when Soviet Union collapsed, he and his buddy went out to Russia, they brought back these half-liter bottles of flavored Russian vodka. They had pull tabs on them. No, they're actually not so bad, but they had pull tabs on them. This is a half-liter of 90-proof vodka. You open the thing up, you cannot reseal it. Now, it's okay if you have a group of 10 people like we did. We went through several of them. But generally, it's one person. You know, there's 10 people. Okay, we're in the 30s at the time. Let's get away with that. But in Russia, you're talking about one or maybe two people downing that, and maybe downing a couple more. So yeah, that's Russian mentality for you. So, despite the enormous capacity, thanks to hepatocyte growth factor, for livers to regenerate themselves, there is a limit. Okay, so that's another one. Now, another kind that many use this particular pathway as well are called collectively the neurotrophins. Okay, neurotrophins stimulate differentiation and maintenance of neurons, especially in the central nervous system. One of the best known examples is something called nerve growth factor. Now, it stimulates neuronal differentiation into mature nerve cells. Now, mature nerve cells are so hyper-specialized that they can no longer divide. They're just so super specialized. But nerve growth factor is required to keep these guys alive. It's produced by glial cells and things like that in the central nervous system. Without nerve growth factor, mature neurons actually die. Okay, well, some interest in some of these things. the possibility some of these neurotrophins, can we use those clinically sometime in the future to repair damage to the central nervous system? Now, some years ago, some lab did a rather interesting experiment. Sounds cruel at first, but you can understand the justification for it. They took a bunch of mice, and they severed the spinal cords of these mice. And of course, the mice were paralyzed from the cut all the way down. The control mice, they gave a fake treatment. The experimental mice, they treated with solutions full of nerve growth factor and other neurotrophins. After a while, the spinal cord on the treated mice partially regrew. Those mice who were totally paralyzed, the control mice were completely paralyzed from that area down. Their hind legs were useless, they were just floppy and dragged around, stuff like that. The experimental mice could actually haltingly walk. Not very well, but in human terms, that's the difference from spending the rest of your life in a wheelchair to maybe walking with a walker or a cane or something like that. So there's interest in that type of research because the thing is, could we possibly repair spinal cord injuries somewhere in the future or partially repair it with that? Yes?
Where did they extract the neurotrophins from to be able to inject?
Well, my guess is they probably use recombinant stuff. In other words, they got the gene for these, and then they just, because these proteins are present in such tiny, tiny quantities that biochemically purifying them is tremendous. You may have to go through kilos of tissue to get nanograms, literally, of the appropriate proteins. So, but once they do that, if they can find the gene, if they identify the protein, they find the gene, they put that gene in bacteria or yeast or whatever and get it to produce. A lot of these protein biotechnology drugs are actually recombinant, including some of the ones I'm getting. That way, you get bacteria to produce it, they can produce in milligram, even gram quantities. So it was probably recombinant stuff. Because a lot of these things, they find the protein, they say, oh, this is great, but it's impractical to get enough to use clinically. But then the biotech companies splice the genes into some kind of organism and then see if they can produce the stuff. So yeah, but anyway, that was an interesting kind of thing, partial regrowth of a severed spinal cord. So, you know, potential, it hasn't come in human clinical trials yet, but the potential of repairing a lot of these spinal cord injuries to some extent is good. It's not going to bring everything all the way back, but, you know, it's better to be able to walk with a walker or whatever than being stuck in a wheelchair for the rest of your life. And, you know, there's tens of thousands of people a year that get nasty spinal cord injuries. Car wrecks, somebody just before I came in here to JSU, was a student at a party decided to dive in the swimming pool without looking to see whether there was any water in the pool or not. So he dove off the deep end, broke his neck, and he was quadriplegic from then on. And you know, places like LA and parts of Birmingham for that matter, and Anniston, you know, people are shooting each other all the time. A lot of people end up 20 years old and they're in a wheelchair because somebody pumped a 9mm round into their spinal cord. So yeah, there's a lot of potential use for these things if we can figure out how to do it, how to apply that to humans. So clearly, these types of growth factors are very, very important. Now, there are many other growth factors around I haven't mentioned. These happen to use the RAS-MAPK pathway, but there are other growth factors that use different pathways, which we will mention further on in the course. Now, at this point, I'll have to admit, there's not enough time to cover through the RAS-MAPK pathway. So I think this is probably a good stop point rather than going halfway through the pathway. So we'll pick this up on Tuesday again, and I will see you folks